had an operation, so he's resting in hospital at the moment. But luckily, Colin's recording this, so thanks, Dad, for all your help all the way. Thanks, Mum, of course. And they got me out on dive boats and doing marine biology since the age of about, well, 1, I suppose. I was brought up on the beaches of the Hoe here, and yeah, I was out on dive trips with the Plymouth Sabbatical Club from that age onwards with my sister, who's also in the audience as well. So I feel like I really do know Plymouth Sound and its marine biology very, very well. But growing up as a diver, basically I was trying to get to warm places, so this is probably my best days. It's some of the biggest fish there are. But getting jobs in marine biology is pretty difficult, and I mainly ended up doing this, which is cold places. So a lot of it is rather unglamorous and chilly and so forth. But you can find out interesting things in these places because most people don't want to go to survey them. They want to hang around in the tropics. Now, I'd like to thank this guy actually, because he's the chap that really got me properly into marine biology. Instead of just messing about going diving and observing things, he got me into taxonomy and the nitty-gritty of marine ecology. Jeff Moore, he's up at the Millport Marine Station up in Scotland, which some of you might have heard of, and he's going through a bad time at the moment because it's facing government cuts now that London University has pulled the plug on funding it. I really hope it survives this period because it's the sort of place where you cut your teeth as a marine biologist. They're in the water all the time, either on boats or under the water, seeing what's out there. And I think that's what this talk is going to be mainly about, is the need for people to get out there and see what's happening, because otherwise I think they're lost. We're not going to know what's going on, just based on computer models or whatever. So this chap was very patient with me. I did a PhD that took a long time, so I was often off going to look at whale sharks and things. And he set up, and he instilled in me the need to collaborate with other people around the world. There was a series of projects that we did with those institutions, looking at the effects, for example, of aquaculture on the seabed, or the effects of fishing on the seabed. And that's what I ended up doing for about 5 or 6 years. And I'll talk you through that because we did find out some interesting things. Now, Jeff is the second from the right there, was the first to do marine biological surveys on Rockall, which is that rock with somebody stood on the top. And it's a piece of land that's way out to the west coast of Scotland. I've tried several times to get there and failed, but I know Keith Hiscock has managed once or twice or several times.
June 1988, I did 3 dives and obviously climbed the rock.
Yeah, very impressive. Very difficult place to get to, but worth getting there because you see unusual things. I think they were being circled by thresher sharks, which is not what you might expect off the west coast of Scotland. So this is what we ended up mainly focusing on, which seems like a weird thing to end up doing, but it's a stuff called merle, which I'd like to pass around, if you don't mind, which usually grows attached to things. So there's a plastic fork. This coralline algae that grows about 1 millimetre per year is encrusting that fork, and sometimes it grows free living on the seabed. And it's pretty important stuff because when we take core samples of the seabed in this living gravel and compare it to mud or sand, it's always got much, much higher levels of biodiversity and is much more rich in terms of the biomass of what's in there. And part of the reason for that is it exudes a chemical that triggers the metamorphosis of planktonic forms. So things like scallops are triggered to metamorphose out of the water column and settle in these areas. So it's kind of important to protect these sorts of areas because they've got chemicals that allow commercial species of organisms to settle and grow. And the seabed generally is important because if you consider all the myriad types of plankton there are on the sea, or all of the dolphins, or all of the whales, or all of the tuna fish and so forth, and add them all together, there is nothing compared to the diversity of what lives on the seabed. 98% of species found in the sea live on the seabed. So the seabed is an important place. And when you start looking in this merle stuff in detail, you find that there's all sorts of levels of organisms that are there, hiding in amongst the interstices of the space that's available. That snail had never been recorded in Scotland until we started looking in the merle. You get organisms that make a living by just shoveling in food and digesting, shoveling in sediment and digesting everything out of it, like that art urchin there, or things with very delicate feeding apparatus that feed on the detritus that's falling down from the water column above. So those are deposit feeders. You get lots of those. You get filter feeders as well. Crinoids, these feather stars, once thought to be extinct and only present in the fossil record, are actually really common as juveniles in these male habitats, and as they grow up, as they get bigger, they then migrate onto rocky substrata. That is my favourite bivalve present in the fossil record. They're actually really common as juveniles in these male habitats, and as they grow up, as they get bigger, they then migrate onto rocky substrata. That is my favourite bivalve in British waters. It's called a flame shell, and it's got these really long orange tentacles and able to swim. And it's got acid in those tentacles to stop cod eating it, and it builds nests on the seabed. So it's really interesting and beautiful to look at, and it usually lives in association with these male bed habitats. And then you've got all sorts of predators that live on the seabed in these areas as well. So I want to try to explain really that there's lots of types of organisms that rely on this habitat to complete their life histories, and that's why we need to look after those habitat types. Now, one of the things we were doing, Jeff Moore and I, was applying to the European Union for funding to see what types of fishing gear were damaging and what types weren't damaging. Because, for example, scallop fishermen were under the impression that scallop dredging these habitats with the merle was really good for the seabed. It was like ploughing the seabed. You break up some people snigger of it. That's what they genuinely thought, because they'd go back to these areas and see live merle again and again. And they thought by breaking it up, they were probably seeding a large area with this material. So what we needed to do was attach camera equipment to scallop dredges, for example, after trawls, hydraulic gear, lobster pots and other things, to see what the relative impacts of the different sorts of fishing equipment were. And well, anyone who's dived in the tracks of scallop dredges will know that this isn't just ploughing the seabed and helping it regenerate, it's smashing it to smithereens. And it's burying and killing these merle-type forms that require light. Anyone who's dived in the tracks of scallop dredges will know that this isn't just ploughing the seabed and helping it regenerate, it's smashing it to smithereens. And it's burying and killing these merle-type forms that require light to grow, and they grow very, very slowly. So if you bury that stuff, you kill it, and you kill it for good, because it doesn't come back, at least not within timescales that are relevant to mankind. So most of the merle habitats that occur in the Clyde, where Milportbury Station, are in fact dead relic deposits, because scallop dredging was invented in the Clyde, and it's been going on since the 1940s. Now, luckily, people listen to this sort of information, and there are now places around the UK where scallop dredging is banned, and it's based on people going out, finding out what this stuff does to the seabed, reporting back to government, DEFRA mainly, and then them trying to think, well, what can we do about this? And famously, there's Lyme Bay up the coast here, off Lyme Regis, where scallop dredging is now outlawed, because we know it does such an incredible amount of damage to long-lived, slow-growing habitat types. Up until, and this is why I think I've become successful as a marine biologist, because I'm trying to get information to the people that can use it to change policy. And I think that's a role that I enjoy, rather than just finding out things for the sake of it because they're interesting. So, for example, in 2007 it was government policy to move fish farms away from areas with small, gentle current flow, because they knew it had a detrimental effect to the seabed, and actively move them into areas with strong water flow. And they asked us at Plymouth University whether we thought that was a good idea, and whether we could do some surveys to find out what actually happened when these fish farms were moved into areas of strong tidal flow. Because their advice they'd been getting from their scientists, who used models and computers to predict where the feces from the fish and the food that they feed the fish would settle on the seabed, they predicted there would be no problem outside the immediate area of the fish farm. So we went to places like in Uist, Shetland and Orkney to find out what was going on in the seabed. So there's a fish farm. Oh, we didn't turn the sound on actually. Yeah, it's better. we're laying down cables on the seabed to find out what's down there. And you see that white stuff there? That's all bacteria. And the bacteria is there because it's being fed by the feces of the food from the farm, and it's turned the entire area anoxic. So when you put your hand into the seabed, you see hundreds and hundreds and hundreds of piled up dead crabs. So the models are predicting, the computer models are predicting, that should not and would not happen. But when you go there, to go to these places, you find out it certainly is. In fact, you have to disinfect your dry suit afterwards because you stink of hydrogen sulfide. It's really, really revolting. So this is another good reason for getting near to these places to see what's happening. And again, SEPA, the Scottish Environment Protection Agency, when they saw this video stuff, they didn't wait for the papers to come out. They said, oh, okay, perhaps our models are wrong, and they've changed their policy. They're not going to put fish farms on top of long-lived, slow-growing habitat types like those flame shelf beds or the mearl beds, which is good news.